Greetings! Thank you for tuning in to this virtual presentation. My name is Mark Bollom, and today I will tell you about the research that I've been doing with bigels and their ability to protect probiotics during in vitro digestion. This research was conducted within the Department of Food Science and Human Nutrition at Iowa State University. To begin, imagine yourself walking through the grocery store and coming upon the yogurt aisle. Upon further examination of some of the packages, you might notice a few terms, such as Bifidus regularis or live and active cultures. Both of these are in reference to probiotics. A probiotic is a live microorganism which, when administered in an adequate dose, confers a health benefit upon the host. Probiotics, as the definition suggests, have many purported health benefits. Many of those are listed here on the screen. However, ensuring probiotics remain alive and are able to confer these health benefits is a challenge for many researchers. Specifically, keeping them alive during processing, shelf life, and digestion are all areas of concern. We saw an opportunity in the food industry to better develop a method for protecting probiotics. Specifically, we were drawn to a bigel. A bigel is a relatively new technology that was first developed in 2008. It is a biphasic system, meaning it has two phases. What differentiates it from other biphasic systems, such as an emulsion, is the fact that both phases are structured or solid-like. Your structured organic phase is called an organogel, or oleogel if edible, and your structured water phase is called a hydrogel. Bigels are unique because they have a diversity in synthesis options. You can use many combinations of gelators, and you can modify your gelation status of both phases, so you can have one phase be gelled, the other one not, or you can have them both be gelled when you homogenize, or you can have them neither be gelled when you homogenize. Additionally, you can modify your homogenization conditions, such as your homogenization shear level, time, and temperature. Now that you know what a bigel is, you might be wondering, why should I use one? First, bigels have been used successfully in some papers to deliver drugs, and so if they can be used to deliver drugs, we're thinking perhaps we can use them to deliver bioactives. Specifically, we're interested in probiotics for dairy applications. As I mentioned, you can use a wide variety of gelators, and potentially you could use one that is a protein. So this gives you the opportunity to increase the protein content of your food, which is going to be on trend. Furthermore, you could use a dairy-based protein, which will increase your dairy content, such as if you were going to implement the bigel into yogurt. And not only would this increase your protein content, but it would offer additional flavor notes that are dairy-based to help mask any off flavors from the oil. Another advantage of a bigel is that they're going to be semi-solid. Additionally, you can manipulate the final characteristics of a bigel using many of the techniques that I mentioned on the previous slide, such as modifying the gelation status, different gelators, or homogenization conditions. Finally, with a bigel, you have the opportunity to have both an aqueous and organic phase. In our case, it gives you the opportunity to have your aqueous phase for probiotics and your organic phase for phospholipids. So why are we interested in phospholipids? Recent work that was conducted in our lab, the publication is forthcoming, found that probiotics in an oleogel with phospholipids had improved viability over probiotics in an oleogel without phospholipids. This test was conducted over a 6-week shelf life study. As stated in the definition of probiotic put forth by the World Health Organization, a probiotic is a live microorganism. We hypothesized that phospholipids may be an effective tool for increasing the viability of probiotics so that the microorganisms can stay alive longer and therefore confer greater health benefits upon the host. We were interested in exploring what effect phospholipids had on probiotic survival during digestion. We knew already from the previous work in our lab that they were effective over shelf life, but it still remains unknown what effect, if any, they have over digestion. We had two main objectives for this study. First, we wanted to determine if a bigel is an effective tool for protecting probiotics during in vitro digestion. Second, we wanted to determine what effect, if any, phospholipids have on probiotic viability within a bigel during in vitro digestion. Our organic phase was in fact an oleogel emulsion. It was composed of soybean oil, soy lecithin, stearic acid, and milk. The milk was where the probiotics were added. Our hydrogel phase was whey protein concentrate 80, and we homogenized the phases together to make a bigel. The ratio of oleogel emulsion to hydrogel was 8:2. Homogenization occurred in 2 steps. First, at a high shear, high temperature. This was done to create a smooth, homogeneous mixture. And then, after the internal temperature of the bigel reached 55 degrees Celsius, we homogenized in the probiotics. Our probiotics of choice were Lactobacillus acidophilus and Bifidobacterium lactis. Now that you know how our bigels are assembled, I can explain how we designed our experiment. As a reminder, our 2 objectives were first, to assess the ability of a bigel to protect probiotics during in vitro digestion, and second, to assess the ability of phospholipids to enhance probiotic viability during digestion. To begin, we have our standard gel, which has both soy lecithin and stearic acid as our organogelators, and then whey protein concentrate as our hydrogelator. Soy lecithin is serving as our phospholipid source here. For our phospholipid-free source, we use just stearic acid as our organogelator and then whey protein concentrate as our hydrogelator. In this case, we only have structure but no phospholipids because there is no soy lecithin. And then finally, our control has neither structure nor phospholipids. The way that we ensured that our structure was as similar as possible between our standard gel and our phospholipid-free one was by doing some pre-experimental work to find at what stearic acid concentration we were able to get an equal firmness value at 37 degrees Celsius between our standard gel and phospholipid-free gel. We chose 37 degrees Celsius as the temperature of our assessment because that is the temperature of digestion. In vitro digestion was conducted using the InfoGest standardized method. Our salivary phase had various salivary salts in it. Gastric phase had pepsin, various gastric salts, and it was acidic. And our intestinal phase had various intestinal salts, bile, pancreatin, and more neutral conditions. We pulled samples throughout to measure probiotic survival. We also did gas chromatography to assess lipolysis to ensure that our enzymes were working and that fat was indeed being broken down. After pulling samples from digestion, our 2 probiotics had to be grown on agar to assess their survival. Lactobacillus acidophilus was grown on M17 and Bifidobacterium lactis on MRS. The specific growth conditions are shown here on the slide. Moving into our results now, we show our lipolysis results on this slide. We have time during the intestinal phase on the x-axis and percent lipolysis on the y. We only measured lipolysis during the intestinal phase since that is where lipase is. What I hope you'll observe is that as digestion progresses, the percent lipolysis increases. This is good news because it means that our enzymes are working and that fat is being broken down. However, something interesting we can do is we can compare the starting amount of lipolysis with the ending amount and see which system underwent the greatest lipolysis. It turns out that our control undergoes the greatest amount of lipolysis, and this makes sense because our control has no structure to limit the ability of the lipase to penetrate into the system and break the fat down. And now the moment you've all been waiting for, do the bigels actually work to protect probiotics? The answer is yes, they do. Our Bifidobacterium lactis is on the left, with Lactobacillus acidophilus on the right, time during digestion on the x-axis, and percent probiotic survival on the y. Beginning with our control in the Bifido did not even survive homogenization, whereas our Lactobacillus acidophilus in the control did survive homogenization. However, upon reaching the gastric phase, they all died. This suggests that gelators serve an important purpose not only during digestion, but also during homogenization. Looking at our bigels now, our Bifido had between 2 and 10% survival, whereas our Lactobacillus were a little bit higher from 30 to 50%. This suggests that Lactobacillus acidophilus is more resilient in the face of adverse conditions of digestion, We believe that our Bigel is an effective tool for protecting probiotics due to its solid-like behavior, as well as protein, which may act as a buffer of sorts. Looking at the effect of phospholipids, our gel with phospholipids had improved viability over the gel without phospholipids. However, the difference was not significant. This is likely because the phospholipids are going to be broken down by the lipase, similar to other fats. And we believe that phospholipids need to be in their native form in order for them to confer protection to the probiotics. In conclusion, we found that our control undergoes the greatest amount of lipolysis due to the fact that it does not have any structure limiting the ability of lipase to penetrate in. Additionally, we found that a biogel is an effective tool for protecting probiotics during in vitro digestion, and we found that phospholipids were not effective at increasing probiotic viability during digestion. Research is not possible without the assistance of others. We would like to thank our funding sources, ingredient donor, and faculty assistants with various components of the project. If you have any questions, please do not hesitate to reach out to any of the authors at their emails listed on this slide. Thank you for listening.